Hello, and welcome to the lecture on viruses. In our virus lecture today, we are going to identify the components and acellular structures of a virus. We'll identify viral replication and how it's different in bacteria, viruses, and animal viruses. That will tell us the terms of prophage and latency, and we'll compare and contrast viral families, and that will be according to their DNA or RNA content. Then we can discuss evidence supporting the correlation between some viruses and cancer. Uh, we'll see how that's especially true with those like HPV. And then we'll look at our friends the prions again and how they differ from viruses. And then we'll also see and discuss some ways we can use viruses in gene therapy. To begin, we'll talk about what we think viruses are. Um, Take a moment in this video to think about the viruses that have affected us recently, namely COVID-19, that we can see over here and in my background behind here. Just how bad diseases and viruses can get. Viruses in particular can spread throughout the world, and they do it through various molecular structures that they have on them, like their spikes, and then inside they have special machinery in their DNA to hijack eukaryotic cells. So besides COVID-19, if you know of any viruses that you may have experienced yourself, put you on your butt for about a week or so. If you've had the influenza virus, you probably plowed through some rhinovirus sicknesses, and maybe you know some other people with some other viruses. And we take that for granted, that knowledge for granted. It wasn't until the 17th century that Jenner even thought of devising a way to inoculate against a pathogen. And so he found milkmaids. We discussed Jenner a little bit. He found that the milkmaids weren't getting smallpox if they had gotten cowpox. So he took it upon himself to inoculate people with with cowpox and prevent smallpox. And he became the father of inoculation. And then we kind of see in the 18th century um, the cowpox and the smallpox thing. Um, but it wasn't until the 1930s that we actually can visualize what a virus is, because we actually need an electron microscope to see them. Remember, we talked about the scale of microbiology. We'll touch on it one more time here. And so uh, they are much, much smaller than bacteria. So that's how you can tell them apart. Same how you can tell a bacteria from a eukaryote. So all viruses share at least these four different traits. Okay, so they are always infectious acellular pathogens, and they don't consist of cells. Their sole purpose is to replicate. They don't think, they don't try to do anything. That just their DNA just is going to replicate. That's what it's going to do. It's going to be able to make these proteins and find another cell and become a parasite within it. So it's going to be an obligate intracellular parasite. Um, Why is it a parasite? Because it actually lacks ribosomes. So since it lacks ribosomes, it can't make any protein. No protein means it can't keep making its protein coats, and it can't keep dividing and going on. So it hijacks the eukaryote, the ribosomes inside of the prokaryotes and the eukaryotes, and sends it these messages to where it kind of hijacks the machinery that the cell wants to uh, output those proteins over its own. All right, and that means in order to do that, it has its own DNA or viral or RNA viral genome. Okay, so that means it has at least the DNA or RNA necessary to keep making the cell over and over again, but it needs the machines inside of another cell to do it. So those is a virus. And finally, in this section of a video, we will discuss the viral scale uh, and the scale of microbiology. And as you can see, where viruses are, we are in the realm of needing an electron microscope. Remember, the electron microscope has a TEM, SEM, and that's how we can see bacteria in those nice 3D images. I think it's still pretty difficult for us to see viruses in 3D, and so most of those are TEM, a 2D image that you see, and you can kind of see some here. And remember, we're about a whole tenfold size difference, maybe even a hundredfold size difference from some bacteria to viruses. So a lot of viruses can surround a bacteria, and even more viruses can surround a eukaryote. Okay, and next we'll go into host and transmission and end the video. So with host range and transmission, it's important to realize that viruses can infect every type of host cell that you can think of: plants, cells, animal cells, bacteria cells, and even archaea. Okay. The best part about it is that most viruses have a host range, though, right? They can only infect certain cells and only a few species of organisms in those species. Okay, so uh, only specific cell types within specific hosts. Okay, that is the way viruses are kept in check. One case in point is our friend the bacteriophage. The bacteriophage is a virus that can only infect bacteria, and it looks like a lunar lander. Okay, you might hear the term called bacteriophage or just phage instead of phage. Um, but know that either way, that this is what infects a bacteria and should not uh, infect humans. Um, so much so that the gene therapy that the viral therapy that we were talking about, could use phage in food, and you can have phage on that targets Listeria bacteria, and put that on some bologna and all your processed meat goods, and it would have to be labeled as such. But the theory is that it could prevent Listeria outbreaks, or you know, uh, a baby who got infected with Listeria because of eating raw meat. So pros and cons for both. I'm not sure if I would eat phage covered food. What would you do? Take a moment and kind of think about that. Okay, so before we end the video, just kind of reiterate our arthropods and how they can spread um, and how they become vectors, how they spread diseases. Okay, they can spread all sorts of diseases, bacteria, protists, and even our friends the viruses in this lecture. And they can function as either mechanical vectors or biological vectors. Mechanical vectors are vectors that aren't included in the life cycle of any specific arthropod. So if it just kind of picked up and moved one area to another, that would be a mechanical vector. A biological vector, though, has a life cycle inside of arthropod. So it grows inside its gut, usually matures, and then gets transmitted through a bite. Okay, and so when you have an arthropod spread virus, it's called arthropod-borne. Arthropod-borne, okay, virus, and that would be an arbovirus. All right, so you would want to know arbovirus. I studied arboviruses in my graduate program, and they are a pretty big topic. Um, Malaria, African sleeping sickness, we'll see in the lab as two cases of arthropod-borne spread diseases. All right, so join me in the next lecture, and we'll continue our talk on viruses.